And trust me, you're probably going to want to get those because it does make a difference in, understanding the stuff that we're going to cover today. And we are going to cover a lot today. So those of the people that aren't here today are going to have to go back and watch this recorded Google Meet in order to get the notes. Otherwise, you'll be very confused if you miss today when I see you guys on Friday. Okay. So again, you're going to need your notes out. You're going to need black or just a pencil, red, blue, and green. Megan, this is on the Unit 5 note packet. Did you pick up your handouts from the front office? Hi, kitty. Okay, so this is Unit 5, Cell Reproduction. This is page 3. Okay, before we begin though, we're going to take a little bit of a second to review because we did a little bit on Wednesday and on Monday, and I had you guys start this ABC review. And we didn't get a lot of vocab down, but here's what I need you to do. So in the chat, you guys are going to list 2 words for me. And I have a list here. These are the vocab words that I covered that I mentioned last class. And I said I'm going to start using them quite a bit. So we need to get an understanding of them. So I want you to pick 2 words that are the most unfamiliar to you. So chromatin, what's a copied chromosome versus uncopied? Diploid, haploid, gametes, somatic cells. What does 2N mean? N, karyotype, mitosis, meiosis, centromeres, sister chromatids, homologous chromosomes, and GO phase, okay? So I want you to pick 2 words that are the most unfamiliar to you, and you're going to type them in the chat, but just don't hit submit yet. Just type them in there, and I'm going to count down in a second, and then you're going to hit send to do a waterfall in the chat feature. So again, pick 2 words out of this list that I have here. We talked about them last class. Type those 2 words into chat. And then when I count down here in a second, you're going to hit the send on the chat. All right, you got some? All right, 3, 2, 1, hit send for me, please. Okay, so here's what I want you to do next. You guys kind of committed to 2 words here. You said those are unfamiliar to you. So I want you to get into Google Classroom. You're going to click on Monday. You can open up your ABC review there. Again, it made a copy for you, so you can also find that in Google Drive. So for example, if you guys put diploid down, then I just want you to go to that slide and put D is for diploid. And let's say you put then chromatin is your second word, then I want you to type in C is for chromatin. We're not going to take the time to fill out the rest of the stuff, but we're just noting you didn't know those words very well. And so on Friday, we'll take some time to make some slides for those to review those vocab words. I'll give you just a couple minutes to get in and add those words to your ABC review document. And you can find that again in Monday's lesson on Google Classroom. When you have those 2 words in your slide, if you will raise your hand, then I'll know when we're ready to go. Again, don't forget those colored pencils. I'm just going to use like black or a pencil, then red, green, and blue. Or if you have like a fine marker or different colored pens, is fine. All right. So let's flip to our notes to page 3. If you guys don't have your notes where you can write, then I would almost recommend that you pause, print out like the next 3 pages of the notes. You can find those in Google Classroom. And then you can watch the recorded meet that I have here for today. I recorded it from the first block today, which is down here. And then you can watch and you can take notes there, and then you have paper to write on. So I'm just saying, if you don't have your notes, or you might pause and take notes and watch this lesson a different way, because I think you'll get more out of it if you're following along and you guys are writing and drawing as we go. All right, on to our notes to kind of talk about where we left off here. There was another worksheet, it was 5.1, and we drew in the corner of 5.1 a cell. And I told you that that cell was in GO stage. And GO stage, let's make sure we have it written in here, is a stage in a cell's life when it's just doing its job for you. It's filtering your blood, or it's killing bacteria, or it's a skin cell, and so it's blocking UV light. It's just working for you. And cells are in GO phase most of the time. Now, sometimes cells die. And what I'm drawing right here is what we drew on that cell in the corner of worksheet 5.1. On this cell that's in GO phase, I put a receptor protein. Talked about receptor proteins can receive signals. They're needed so cells can communicate. And so this cell in GO phase, there was a cell next to it that died. And that cell released chemicals that can hook on to the receptor protein of the cell and signal this cell to divide, because we need new cells in that area because a cell has died. And so cells should not divide until they get a signal to do so. That's very important. Cells should not divide unless they get a signal to divide. Can anyone tell me what we call cells that are dividing even though they haven't received a signal to divide? There you go, cancer cells. They'll divide even though they have not received a signal to divide. So it's really important that cells don't divide unless there's a need for that, okay? So now that this cell in GO phase got a signal to divide, then this cell is going to enter a new stage of its life. Okay, so we say that this cell is going to enter what's called the cell cycle. So after a cell gets a signal to divide, it's going to enter what's called the cell cycle. And as you can kind of see from the diagram here, the cell cycle is all about changes in the cell so that we end up with 2 cells now. The end result is the cell is going to divide at the end of the cell cycle, okay? All right, now you're gonna see this diagram a lot, and you guys are gonna draw diagrams similar on Fridays. So let's make sure you have a general understanding of what's going on. Oh, by the way, let me go back up here to the second bullet. Remember we talked about there was a couple cells that can never divide to give you new ones. Can someone give me an example of a cell that once it dies, you'll never get a new one back? Yeah, so nerve cells. So if you've ever had surgery and they've had to, like cut into tissue, then odds are they probably cut some nerves. And that scar, that area where the surgery took place and they made that incision is going to be numb forever because those nerve cells in that area died whenever they were cut into. So nerve cells and muscle cells are another type of cell that never can divide. So a heart attack and losing muscle of the heart is forever because muscle cells and nerve cells are too complex to divide. So they are always in GO phase. They can never enter the cell cycle. They can never divide. Okay, So let's take a look at the picture of the cell cycle first and just note a few things. There are three main stages of the cell cycle. But if you notice, there's a lot more than three stages in this diagram. So the three main stages are right here. I'm going to put a number 1 here in the middle. Number 1 is interphase. And you can see that interphase is most of the cell cycle. As a matter of fact, interphase is this whole area of the circle is representing this stage of a cell's life after it enters the cell cycle. So interphase is the first and the longest stage. And then let's put a number 2 here. After interphase, the cell goes into mitosis. And this whole brown region right here, that is representing mitosis. Sometimes they call it the mitotic stage, or M phase stands for mitosis, okay? And then the short little phase here at the very end, that's represented by purple on my screen. Let's put number 3 there. That is cytokinesis, okay? So that's what we're going to write here in these lines. So the three main stages, the first is interphase. And it might help you if you look at your notes here and you copy the words here, because it's going to take me a while to write. So we have interphase, and then we're going to write mitosis. Mitosis is written here. And cytokinesis are the three main stages. Give you a second to write those. All right. So what we're going to do just on this first page is we're just going to talk about, or on this page we're doing now, we're just going to talk about in general what happens in those three phases: interphase, mitosis, and cytokinesis. Okay. So first of all, for interphase, this is what I want you to think of what happens in interphase. Okay. So let's say that you guys are moving into a new house on Saturday. Okay. The moving trucks show up on Saturday. I want you to think of what today, tomorrow, and Friday would look for you, right? You are going to be super busy. You are going to be preparing for division. You're going to be making sure, that, you're packing up your furniture to go into the new house. So it's going to be really busy, but you're just preparing. You're not actually moving the next 3 days. So that's what interphase is all about. So if you want to kind of highlight that here, and I want you to add kind of a star here, is it's preparing. So just kind of put prepare to move. That's what interphase is like. So you're preparing, you're getting stuff ready. So preparing to move. Now, if you look, interphase itself has 3 stages that it's divided into. And you will have to remember those for me. So the first is called G1, which makes sense. After GO, the cell enters a stage, which is called the first stage of the cell cycle or of interphase is called G1. And then after G1 is done, then the cell is going to enter a new stage of its life, which is called S phase. And then finally, the cell is going to go into G2. Okay? And once interphase is over, then it's moving day. It's time to actually move things around in the cell. And that's what's going to happen in mitosis and cytokinesis. Now, all really that mitosis is division of the nucleus. So mitosis sometimes is just called nuclear division. If I say what type of nuclear division is taking place, your answer is either going to be mitosis or meiosis, which we'll talk about a couple class periods from now. So we're dividing up the nucleus, which technically that means that we're dividing up the DNA in those steps of mitosis. And you can see from the diagram here, mitosis itself is actually 4 smaller categories. So it has 4 substages. So you'll want to remember PMAT, for the order of those 4 stages. We have prophase, metaphase is the M, anaphase is the A, and then telophase. It also might help you to remember pro means first, and the thing to remember about telophase is that it is in alphabetical order. T comes last, so that might help you remember telophase is the last stage, PMAT. Okay. Now, the biggest idea to kind of understand a big difference between mitosis and meiosis is that at the end of mitosis, you get TOS. And notice mitosis has that in there, TOS. So I want you to write 2 of same. That's what TOS means, 2 of same. And what I mean by that is that if I tell you that this cell is a diploid cell, do you guys remember the abbreviation for diploid?
Isn't it 2N?
Very good, because di means 2. So our abbreviation for diploid is 2N. So if I tell you that this cell, the 2 in number, is 46, and that would be for humans. And then there's a few other species that have 46. We're not the only ones. We haven't locked in that number as ours. There's other species that have 46 chromosomes. So if this cell gets a signal to divide, and this cell started out with 46 chromosomes, when it's done dividing, you have to be able to tell me, if I ask you how many chromosomes will be in this daughter cell, you would have to say 46. So this is going to be a diploid cell that we get at the end, and so is this one. Okay. So there'll be a lot of number questions on an assessment, and these should be really easy if you remember TOS. If I say that this cell comes from a camel, camels have 70 chromosomes. If a skin cell of a camel divides by mitosis, how many chromosomes will be in each cell? And it should be 70. It should be whatever the species number is, okay? Because it doesn't matter what cell I get from you. If it's a somatic body cell, if it's a human somatic body cell, I will see 46 chromosomes in all those, okay? So anytime a cell divides, we need other cells new cells in your body now that also have 46 chromosomes, okay? So mitosis is going to take care of the nucleus. Cytokinesis, as it kind of suggests with the name there, divides the rest of the stuff, which is basically the stuff that's in the cytoplasm. All right. So again, to summarize, the cell enters the cell cycle, and most of the time, while the cell's in interphase, it's getting ready for division. Mitosis, division started. The nucleus is being divided, and then at the very end, the rest of the contents are going to be divided, and that's the cytokinesis, right? So moving on to the next, and this is where you're going to need those colored pencils. I haven't forgotten about those yet. Okay, so now what we're going to do here is we're going to draw a cell and what it looks like going through the stages of its cell cycle. And this is really similar to like, what if I drew you going through the stages of your life, okay? So we would draw you just later on in time and what you would look like as you go through the different stages of your life. So that's what we are doing on our notes starting on this page. So here I have a cell, and this cell, we're going to say its diploid number is 4, okay? So that means that this cell is supposed to have 4 chromosomes in all of its body cells. Now, sometimes you're going to see the DNA, and then the DNA is going to be shown in chromosome form, like I have here in this cell. So this cell is just another way to show that it has 4 chromosomes, as I can draw 4 chromosomes in it. By the way, are those chromosomes that we're looking at in this starting cell, Would they be copied or uncopied chromosomes?
Uncopied.
Very good, because when they're copied, they look like X's, okay? So if I say draw a cell with 4 uncopied chromosomes, this is what you would draw, okay? But here's the problem. This is where it can get a little bit confusing. Is if this cell is in GO phase, okay, if I can take a picture of what this cell looks like in GO phase with 4 chromosomes, it doesn't really look like this. The DNA is never coiled up in chromosomes until that cell hits mitosis. So the DNA in GO phase, G1 phase, S phase, G2 is actually in chromatin form. And when DNA is in chromatin form, you can't see it. You can't see the you can only see chromosomes. You can't see chromatin, okay? So really what a cell is going to look like if it has 4 chromosomes of DNA and it's in, GO or G1 phase, is the DNA is going to look like this. Okay, so this is where we're going to use our colored pencils. Each chromosome is in chromatin form. It looks like a pile of spaghetti. And so I'm going to draw each chromosome. If my color would work here, let's try that again. So I'm going to draw each chromosome as a small pile of spaghetti. Don't draw a lot of spaghetti, and there's a reason why. Okay. So this is really what the DNA would look like. And if I would zoom in like with a microscope and look at a cell that has entered G1, it would really look like that. It would not look like this. Okay. So the DNA of a cell doesn't ever coil up whenever the DNA hasn't been copied yet. Okay. It doesn't look like that. All right. So now let's get in details. What is happening? The cells entered the cell cycle. What is the cell doing? Okay. Well, first of all, I want you to highlight that the G in G1 can stand for growth. So if you want to circle the G over here, that's one thing that the cell is doing. So notice between G1 and the next stage of its life, that cell got bigger. So the cell grows. Now, in S phase, something really important is going to happen. The DNA is going to get copied. Because remember, we need to take the cell and split it into 2. So we need 2 sets of DNA. So to kind of emphasize that, take this black chromosome and double it. Make it twice as big. Same with the red. Make it a pile of DNA that's twice as big as the original. And again, to kind of practice vocabulary, DNA, when it's not in chromosome form, when it looks like a long string, it's called chromatin. Okay. So that's what S phase is all about. The S phase, or S, stands for synthesis. So why don't you circle the S, and we're going to write in synthesis there. And synthesis means to make. I'm just going to add synthesis here. Synthesis means to make. And so the cell is making an extra copy of the DNA. Now, the fancy scientific name for that you'll need to recognize is called replication. The DNA is being replicated. So DNA replication is occurring. DNA replication is occurring. Okay. If you replicate a picture, that means you just redraw it or you copy it. So replicate means to copy. Now I'm going to go back up here to G1, and I purposely didn't add something in this last blank here because it kind of relates to what's going to happen in the next phase. So imagine if, and you would never do this, right? Imagine if you had an assignment due and you were desperate and you copied someone else's assignment, right? Okay. Now I guarantee you, in that time of desperation, when you go to copy, you probably look at that person's work to make sure it's okay to copy, like it looks good to copy, right? And that's something that's going on in G1 phase. is if the DNA gets copied in the S phase, during G1 phase, the cell is checking and repairing the DNA to make sure it's good to copy, because we don't want to copy bad DNA. If the DNA is bad, or the DNA has been mutated, is what I mean by the DNA is bad, then that could be a cancerous cell. So we want to fix the DNA before we copy it, because mutated DNA could lead us to having a cancerous cell. Okay, so again, to just emphasize, in G1 phase, the cell is growing, it's making some additional organelles, and it's checking, and if there's any mutations in the DNA, it's repairing it. If everything's good with the DNA, the cell goes into the S phase. Then the DNA is actually getting copied or replicated. Okay. After the DNA is done being replicated, the cell is going to move into G2 phase. And this is another important checkover event. So during G2 phase, that cell is making sure that the DNA got copied or replicated correctly. Because this is a big event, copying the DNA. Let me give you an idea of what your cell has to do in a couple hours with its DNA when it's replicating it. Imagine I give you an essay that is 4 billion letters long. Okay, so the letter count is 4 billion letters is the length of this essay. And I asked you to copy that essay word for word, letter for letter. Do you think you might misspell a word or leave out a letter or leave out a word if you had 4 billion letters to copy? Well, that's what the cell has to do. Your DNA is 4 billion nucleotides or bases long. And we've talked about DNA a little bit. We'll talk more about it next unit. So imagine copying 4 billion letters. Now your cell's really good at it. It makes one mistake. It changes one letter in 1 billion letters. Let's put that in context. Imagine you had to do 1 billion math problems and you only messed up and got the answer on one of them. You'd be pretty darn accurate, right? So the cell is incredibly good at replicating the DNA, but it does make mistakes. So almost every time your DNA gets copied, there's 4 mistakes. And your cell in G2 needs to find those mistakes and correct them, okay? All right, so that's an important event, is during G2, the cell is making sure the DNA has been copied correctly, okay? You might emphasize the G and then emphasize the G, because notice the cell is growing a little bit more, because we're going to split the cell in 2. So we need to have a big cell. Otherwise, we just end up with 2 little tiny cells, okay? And you might note, let's put a star here. And then this is the final, let's indicate, this is like the day before moving. It's chaos. You have a lot of final preparations to make. And so if this is the stage right before we're actually going to start to move stuff, the cell's got to get ready. Checking the DNA. It's going to need proteins to help move things around, so it's making additional proteins. We're going to need double the organelles, because we're going to have 2 daughter cells here from 1, and that cell's got to get a little bit bigger. Okay. All right, once everything checks out and is ready in G2 phase, now it's the next stage is basically the moving day. It is now time for the cell to start dividing. Hasn't even divided yet in those 3 phases. It's just preparing to. And so when that division starts, we say that the cell has moved into the next stage of the cell cycle, which is mitosis. Mitosis is going to help the cell split up the 2 copies of DNA that it made in S phase, okay? Now, as for that cell in G2, we're going to draw the DNA just the same way as we did in S phase. So it got copied in S phase, and now that copied DNA is still in the cell. The copied DNA is being checked over. If there was mistakes made, we hope that cell can find the mutations and fix them. Okay. And if it doesn't, we can have a problem. We could end up with a cell with mutated DNA. The cell can't fix it. that could be bad news because that DNA can cause that cell to start to divide out of control. All right. Now, it's really hard for me to write with this, and it does take me longer to do. So here's what I'm going to have you guys do to finish up the block. You're going to go into Google Classroom here and click on Wednesday. And notice here you have a YouTube link for basically task number 2. And this is the rest of the notes. Now, I couldn't have you watch the whole video because I actually changed page 3 and page 4 a little bit, and it wouldn't match what's in the video. So that's why I want you to open this YouTube and fast forward to basically 9 minutes and 50 seconds. And then you're going to start the notes from there and complete the next page and a fourth of notes using the video. There's a lot that has to be drawn. And like I said, you can go faster and notes can go faster if you guys watch the video. So that's one thing I want you to finish. The second thing that I need you to do is you're going to click on this link here that says Mitosis Mover. And there is a interactive, and it's also a bit of a quiz that you guys are going to finish. When you get done with this interactive, it's going to ask you what to do with your results. So as you are learning and reviewing and taking this quiz, up here in the corner, you're going to see a percent for how many of the questions you've gotten right. And when you're done, I think the screen shows it, then you have a choice to email me or do a print screen. You're just going to email your results to me. And again, my email address is mspencer@gbr5.net. I do have that here in Google Classroom if you forget what my email is. Or I'll put it in here. It doesn't look like I have it, but I'll put it in there for you, okay? But again, before you start this, I just want you to note that when you answer these questions, these quiz questions as you go, most of the answer is in the reading that they're expecting you to do here, okay? So whenever you start this, then it's going to pop up some text. You need to read to learn and read to review because it's going to basically go over everything that I kind of went over here in your notes to see if you got it and if you understand it. And if not, like I said, it's teaching you as you go. Don't rush through it. Even if you don't rush through it, it's only going to take you about 6 minutes to do. So again, read and complete this to understand, not to just get it done. And email me those results when you're done. Okay? All right. I am not going to talk to you probably for the rest of the block, but I am going to stay on here. So if you guys have any questions or you're having issues and you don't know where to go, then you can send me something in chat or you can just unmute and ask me. But again, all you need to do is watch the rest of the lecture, start at 9 minutes and 50 seconds, complete the Mitosis Mover, email me your results, and have a great snow day. Thank you.